Hi everyone and welcome to my YouTube channel. Today we will be talking about the introduction to genetics. How did this field of science has emerged over time. So you can see in this photo the structure of an overview of the DNA. Okay, and DNA is just one of the mightiest molecule that we will be looking for when we study the field of genetics. Let's start. So there are newer model organisms in genetics. So these include your roundworm here, your Caenorhabditis elegans, your zebrafish, the Neorerio, and your mustard plant, which is your Arabidopsis thaliana. So these model organisms are which the geneticists are using, okay, because they have almost the same or almost related sequence for humans. And when we study genetic testing, okay, these animals were being utilized in the laboratories. So what are the chapter concepts for this lesson? So we all know that genetics is the study of information processing or the transfer of information from each generation in your living organisms, and it is very rich in tradition of discovery and experimentation, which stretching from the ancient world through the 19th century to the present day. And one of the good subdisciplines of genetics is what we call as your transmission genetics, which is a general process by which traits controlled by genes are transmitted through the gametes from generation to generation. And this is a process, or the cell division process, we call it meiosis, cell division, which is the generation of your sex cells or that. Also, mutant strains can be used in genetic process to map the location and distance between genes on your chromosomes. This is also important to note that Watson Crick model of DNA structure explains how your genetic information is stored and expressed using that double helical structure with the nitrogenous base pairs. And the discovery is the foundation of molecular genetics, another field of molecular genetics. Recombinant DNA technology revolutionized genetics, and that was the foundation for the Human Genome Project, which was found in the late 2000s, 2003. This Human Genome Project has generated new fields and combining genetics with information technology. And next would be biotechnology. It provides genetically modified organisms, or what we call as your GMOs, and their products that are very used across a wide range of fields, including agriculture, medicine, and industry. Some model organisms are now used in genetics, and in research, they are utilized in combination with recombinant DNA technology and genomics to study human diseases. And genetic technology is quite developing faster than the policies, laws, and convention that govern its use. So let's start with this very phenomenal molecule, and we call it your Crispr Cas. Okay, your Crispr Cas is a molecular complex which are found in bacteria and that has the potential to revolutionize our ability to rewrite the DNA sequence of genes from any organisms, saying us that we could technically we could customize babies or design babies in accordance with what the preferences of the parents. Okay, so you can see the photo or the diagram on your right, a crystal structure of your CRISPR RNA guided surveillance complex. So we will be looking and talking about CRISPR in the next few slides. Another important breakthrough in genetics are systems that have been developed, including your zinc finger nucleases or ZFNs and your transcription activator effector nucleases. Okay, so these are your ZFNs are artificial restriction enzymes which are generated by fusing a zinc finger DNA binding domain to a DNA cleavage domain. So these zinc finger domains are can be engineered to be to target specific desired DNA sequences, and this enables your zinc finger nucleases to target unique sequences within your complex genome. And as the process go on, taking advantage by this endogenous DNA repair machinery, okay, we are able to use reagents, okay, to precisely alter the genomes of higher organisms. So this example is a ZFN process, okay, a pair of ZFN with 3 zinc fingers here, okay, and they are bind to a target DNA, and they are shown, okay, introducing a double strand break. So at the FOKL, or the focal domain, which are depicted in yellow, this one, subsequently the double strand break is shown being repaired through either homology directed repair or non-homologous end joining. So this this healing process, okay, is resulting into the targeted gene replacement or your targeted mutagenesis. Another concept is your transcription activator like effector nucleases. As you can see the photo on your diagram on your right, okay, this is a specific drawing of a dimeric tail focal fusion. The blue, okay, is the tail, okay, the green is your focal, which is bound to your DNA, colored in orange. So going back to your Crispr Cas, okay, Crispr Cas is highly phenomenal, okay, because they were initially discovered as a molecular complex that protects bacterial cells from invasion by viruses, or the what we call as your bacteriophage. CRISPR, or shall we say, is an antonym for clustered, regularly interspersed short palindromic repeats, which is designating an RNA molecule which in the laboratory can be synthesized to match any DNA sequence of choice. So your CRISPR RNA has 2 ends. One that recognizes and binds to the matching DNA sequence in the gene of interest, and the other is binding to a CRISPR-associated or Cas nuclease or a DNA cutting enzyme. So you can see of the diagram on your left is a secondary structure image for the CRISPR DR2 RFO1315, which the nucleotide coloring indicates the sequence conservation between the members of this family, okay, and with the red end spectrum labeling highest conservation. So CRISPR-Cas is highly phenomenal because in the US, a clinical trial using CRISPR-Cas9 for genome editing in cancer therapy has been approved, and second proposal for treating a genetic form of blindness is in preparation. And there are some many diseases that are easy to treat because of these genetic editing mechanisms. The photo on your the first photo is the one which is affected with the sickle cell anemia. This one is a hand of a person with severe anemia on your left, and this one is without anemia. This is muscular dystrophy, which is a genetic disorder where the muscle tissue wastes away and loses its function. This one, in your affected muscle in your right, this one, this is an affected muscle of your muscular dystrophy, and you can see that this is quite disorganized, okay, and the concentration of your dystrophin in green is somehow greatly reduced. This is a muscular dystrophy sketch of the disorder when the muscle tissue wastes away. This photo here is a fundus of the patient with what we call as retinitis pigmentosa. It's a mid stage, so you can see a bone spicule shaped pigment deposits which are present in the mid periphery along with your retinal atrophy. So your macula is preserved, although a peripheral ring has a depigmentation. Retinal vessels also are attenuated. And this one is a photo with a healthy airway, and the one which has a obstructed or blocked airway caused by a genetic disease, cystic fibrosis. So these are just some of the diseases that are well treated, okay, with what we call as CRISPR, and some of the genetic breakthroughs that we are having in this century. But before that, we will go back to the interesting and very rich history of genetics. This is a form of a watercolor tracing by archaeologist Henry Broyle from a cave painting of a whole-life canid. Okay, this is 19,000 years ago. Domestication of plants and animals were the highlight of the history of genetics, because people are get acquainted with the ones which they prefer the most. Okay, they wanted these particular traits. That's why I in my presentation I've posted a wolf, okay, like canid, because these are the immediate ancestors, okay, of our modern day dogs, and dogs are domesticated animals. Okay, in this golden age of Greek culture, the writings of the Hippocratic school of medicine in 500 to 400 BC and of the philosopher and naturalist Aristotle discussed heredity as it relates to humans. Okay, the photo on your left is a mural painting showing Galen and Hippocrates in the 12th century. The Hippocratic treatise on the seed argued that there are active humors in various parts of your body, and they served as the bearers of hereditary traits, telling us, okay, that withdrawn from various parts of the male body, okay, to the semen, and it's passed on the offspring. And these, what we call as the humors, okay, could be healthy or somehow deceased, or there is an illness accompanied by it. So these humors accounting for the appearance of the newborns or the congenital disorders or deformities, they will always go back to these humors. Okay, it was also believed before that these humors could be altered in individuals before they were passed on to the offspring, and explaining how newborns could inherit traits that their parents had acquired in response to their environment. So there is the earliest manifestation of your nature and nurture concept. Another story from the Greek culture is that Aristotle extended your Hippocrates thinking and proposed that the male semen is containing a vital heat, and it has the capacity to produce offspring of the same form, and it follows the basic structure and capacities as the parent. Okay? Aristotle believed that this heat cooked and shaped the menstrual blood produced by the females, and they gave rise to the physical substance and produced the offspring. Okay? Although ideas of Hippocrates and Aristotle sound primitive and naive today, we should recall, okay, that prior to the 1800s, neither sperm nor eggs had been observed in mammals. And these people are producing information which they believe, or assumption, or a hypothesis to what they believe in. So in 1600s to 1850s, this is what we call as the dawn of modern biology. In 1600s, William Harvey studied reproduction and development and proposed the theory of epigenesis, and it states that an organism develops from a fertilized egg by a succession of developmental events that eventually transform the egg into the adult. Okay? And the theory of epigenesis directly conflicted with the theory of preformation, which stated that the fertilized egg contains a complete miniature adult, which is called a homunculus. So you can see a photo on your right, a tiny person inside a sperm, which is drawn by Nicholas Hatzroker in 1695, and the homunculus. So they believe that an organism is contained within the gametes, okay, and it will develop as the time passes by. Another theory, okay, which was proposed by Schleiden, Schwann, and Virchow, which is the cell theory, and stating that all organisms are composed of basic structural units, we call them cells, which are derived from pre-existing cells. A photo on your right is a drawing of the structure of cork, okay, from Robert Hooke that appeared in his book Micrographia. Very interesting, okay? From William Harvey to Schleiden and Schwann, the cell theory. Next would be the idea of spontaneous generation. And this is the belief that the creation of living organisms from non-living counterparts was disproved, okay, by Louis Pasteur later in the century, and living organisms were then considered to be derived from pre-existing organisms and to consist of cells. So on this diagram, Aristotle proposed that in sexual reproduction, the child inherits the form. Okay, it is also called the eidos. This is the green part here, and they got it from the mother. Okay, they got it from the father, and also they got it from the mother. Okay, as well as the heat, which is what we call as the pneuma, either from the father, okay, this one, or from the environment. In spontaneous generation, the environment could effectively replace, okay, they will replace the parents' contributions of form, matter, and heat. And that is what Louis Pasteur is believing. In the 1600s to 1850s, the revolutionary work during the mid-1800s, Charles Darwin and Gregor Mendel set the stage for the rapid development of genetics in the 20th and the 21st centuries. And very importantly, this person on your right formulated the theory of natural selection, presented an explanation of the mechanism of the evolutionary change as he went along his HMS Beagle, his expedition on the Galapagos Islands and the different parts of the world. He formulated and proposed natural selection. And another person in the name of Alfred Russel Wallace, okay, noticed that natural selection is based on the observation that populations tend to contain more offspring than the environment can support, and leading to struggle for survival among individuals. And that's the competition that will happen in a certain population where some of organisms are struggling for survival. And this was the theory, or some part of the theory, from Charles Darwin. And Darwin, however, lacked an understanding of the genetic basis of variation and inheritance, and this gap left his theory open to reasonable criticism well into the 20th century. Shortly after Darwin published his book On the Origin of Species, Gregor Johann Mendel, an Augustinian monk, published a paper in 1866 showing how traits were passed from generation to generation in pea plants and offering a general model of how traits are inherited. This is very a big breakthrough in genetics, because his research, Mendel's research, was a little known until it was partially duplicated and brought to light by Karl Correns, Hugo de Vries, and Erich Cermak around 1900s. By the earlier part of the 20th century, it became clear that heredity and development were dependent on genetic information which are residing in the genes contained in your chromosomes, which were then contributed to each individual gametes. Okay, so we called it the chromosomal theory of inheritance, and this gap in Darwin's theory was closed. Okay, and Mendel's research was continuing to serve as the foundation of genetics. And there, genetics progressed from Mendel to DNA in less than a century. Mendel's work on transmission of traits was the highlight of this part because Mendel applied quantitative and data analysis to his results and showing that traits are passed from the parents to offsprings in predictable ways. Because of that ratio that he proposed, and he further controlled that each trait in the plant is controlled by a factor, we called it now genes, and that during the gamete formation, the formation of egg cells or the sperm, and members of gene pair which are separating from each other. That's his law's independent assortment of law of segregation. So you can see the photo on your right is a woman picking peas in Mount Kenya region of Kenya. So Mendel's work has set the foundation for genetics. Genetics, broadly defined as a branch of biology which is concerned with the study of heredity and variation. The chromosome theory of inheritance is uniting Mendel and the microscopic view of genetics, which is the meiosis and mitosis, cell division. About 20 years after his work, advances in microscopy allows researchers to identify chromosomes and to establish that in most eukaryotes, the cells that has the nucleus, members of each species have a characteristic number of chromosomes, and we call that one as the diploid number 2n in most of their cells. And for example, humans have a diploid number of 46. 22 of them are autosomes and one pair of them are the sex chromosomes. Chromosomes in diploid cells exist in pairs and we call them homologous chromosomes. You can see the photo on your right, a human chromosomes during your metaphase phase. And a photo on your left is a colorized image of human chromosomes that have duplicated in preparation for cell division and as visualized using an SEM or a scanning electron microscope. A photo on your right is a colorized image of a human male chromosome and arranged using a karyotype, decreasing order of the length of these chromosomes, and lastly, it will dictate the sex of an organism. A photo on your left is a late stage of mitosis after the chromosomes stained in blue have separated, and a photo on your right is condensing chromosomes, which has an interphase nucleus on your left, condensing chromosomes in the middle, and condensed chromosomes on the right, and they are produced using your anti-dsDNA antibodies using an HEP 2010 cells with a FITC conjugate. So another theory proposed by Boveri and Sellen, chromosome theory, independently noted of the behavior of these chromosomes. The behavior of these chromosomes during the cell division or the meiosis is identical to what the behavior of genes during gamete formation as described by Mendel. For example, genes and chromosomes will exist in pairs, and members of a gene pair and members of a chromosome pair will separate each from each other during the gamete formation, going apart, and that's the theory of segregation and the law of independent assortment by Mendel. Boveri and Sellen independently formulated the chromosome theory of inheritance, stating that inherited traits are controlled by genes which are residing on chromosomes and fatally transmitted through gametes, it's either the sperm or the egg, and maintaining genetic continuity from generations to generations. And these genetic variations is very visible using this chromosome, of your Drosophila melanogaster, the chromosome 1, the X chromosome of one of the sex-determining chromosomes of your Drosophila melanogaster or the fruit fly. And they have located several genes like cute bristles, white eyes, ruby eyes, okay, cross-veinless wings, singed bristles, lozenge eyes, vermilion eyes, and many more. So in your chromosome, okay, your chromosome I, you can see these particular alleles that are found within the arms of your chromosomes, and they can contain hundreds of genes. About the time that the chromosome theory of inheritance was proposed, scientists began studying the inheritance of traits in the fruit fly, Drosophila melanogaster. Okay? And they found genetic variation. You can see the photo on your left in a white-eyed mutation and the normal red color on your right. This variation was produced by mutation in one of the genes controlling, okay, the eye color of your fruit fly. The white eye variant discovered in Drosophila is an allele of a gene that controls eye color, and alleles, as defined, are alternative forms of a gene. Different alleles may produce different or observable features, or we call them as a phenotype of the organisms. Rather, the set of alleles for a given trait carried by the organisms is what we call as your genotype. And using mutant genes as markers, geneticists can map the locations of genes on the chromosomes. And this, in 1944, the search for the chemical nature of genes was finally ended, and if it's a DNA or if it's a protein. Avery, MacLeod, and McCarthy, researchers at the Rockefeller Institute in New York, published experimentations showing that the DNA was the carrier of genetic information in bacteria, and this evidence was clear-cut failed to convince many influential scientists. In this left image, you can see a strands, okay, of purified DNA such as this, which are precipitated from the solutions of cell components to perform some of the bacterial transformations. Another experimentations provided the evidence, okay, for the role of DNA as a carrier of genetic informations came from Hershey and Chase experiment, okay, and they've worked on viruses. So your bacteriophage, okay, virions or viruses that affects or infects the bacteria, they have this sulfur labeled protein in red, and then attaching to the cell, releasing the genetic components into your cell, and they got, okay, the bacteriophages and the result, okay, of this centrifugation with no sulfur in cells. Another thing, and when phosphorus is labeled DNA, okay, in green, because one component of your DNA is phosphorus, when they infect the cell, okay, they got blended and the virions or bacteriophage were removed after centrifugation. Thus, they have noted that the genetic material was remaining inside, or the DNA remained inside your cells. So they've noted that the genetic material is the DNA. And with that, the discovery of your double helix launched the era of your molecular genetics, and the DNA structure was the greatest discoveries done by James Watson and Francis Crick in 1953, and they discovered the structure of your DNA. DNA is a long, ladder-like macromolecule that twists to form a double helix, and each linear strand of the helix is made up of a subunits, we call them nucleotides. You can see this photo, a summary of the structure of your DNA, illustrating the arrangement of the double helix on your left and the chemical components making up each strand on the right. So you will see the dotted lines here. They represent weak bonds, okay, called hydrogen bonds, and holding together the two strands of the DNA helix, and they got their complementary base pairs. Another material of genetic material that they've discovered is your RNA. It's another nucleic acid which is chemically similar to DNA but contains a different sugar. It's a ribose rather than a deoxyribose, and in its nucleotides it contains the nitrogenous base uracil in place of thymine. RNA is, however, a single-stranded molecule. You can see a hairpin loop here from a pre-mRNA. It highlighted are the nucleobases, which are in green. Okay, the ribophosphate backbone, which is in blue, and this is a single-stranded DNA that folds back upon itself. Another discovery was the gene expression, okay, from DNA to phenotype, and they have got into different processes. It undergoes replication, transcription, from DNA to mRNA, translation, mRNA into a series of polypeptides or amino acids, which you call as your polypeptides or proteins. So we can see it here in this diagram. Proteins and all biological function. Most cases, proteins are the end products of gene expression. As we all know, in the central dogma of molecular biology, the polypeptide chains are what we call as proteins, and they have certain biological functions that they perform. And the diversity of life itself, okay, arises from the fact that proteins, okay, are made up of the combination from 20 different amino acids. Exclusion of that will be your nonsense codons, the UAG, UAG, UAA, and UGA. And very unique amino acid, your phenylalanine, okay, and the methionine, which will be the start codon. And all of these information will be discussed in the video, the genetic code, transcription and translation. So this is a crystal structure of chaperonin, a huge protein complex. A single protein is highlighted, and chaperones assist protein folding. Enzymes, okay, the largest category of proteins, and these molecules will serve as your biological catalyst. It lowers the energy activations in reactions and allowing cellular metabolisms to proceed at body temperature. So you can see this structure, a coenzyme folic acid on your left, and the anti-cancer drug methotrexate on your right. A very similar in structure, this green part here. As a result, your methotrexate is a competitive inhibitor of many enzymes that are using your folates. The lock and key hypothesis. These are your enzymes. Generally, they end in ASE. And proteins and other enzymes, they are very critical components of your cell and other organisms. They include hemoglobin, which is an oxygen-binding molecule in your red blood cells, insulin, a pancreatic hormone, which regulates glucose levels. Collagen is a connective tissue molecule, which is found in your skin, okay, your dermis. Actin and myosins, which are the contractile muscle proteins, which helps the relaxation and contraction of your muscle fibers. So you can see a photo on your right, a hemoglobin concentration measurement, which is administered before a blood donation in your American Red Cross Boston Blood Donation Center. So we will now link genotypes to phenotypes. In this case, we will be focusing on a disease. We call it the sickle cell anemia. A sickle cell anemia is caused by a mutant form of hemoglobin, a protein that transports oxygen from lungs to the cells of your body. And hemoglobin is a composite molecule. It is made up of two different proteins, your alpha globin and your beta globin, each encoded by a different gene. And in sickle cell anemia, the mutation in the gene encoding your beta globin causes an amino acid substitution in one of the 146 amino acids in the protein. Look at this. This is your normal beta globin and the mutant beta globin. Okay, instead of producing this amino acid, amino acid, sixth amino acid, it produces a different amino acid, is producing a valine. Okay, so a single nucleotide change, okay, in your DNA encoding the beta globin, leading, okay, to a change or altered state of your mRNA codon, and insertion of a different amino acid, okay, that's producing your sickle cell anemia. And individuals with 2 mutant copies of the beta globin gene will have a sickle cell anemia. Their mutant beta globin proteins will cause the hemoglobin molecules in your red blood cells to polymerize when the blood's O2 levels are too low, forming a long chain of hemoglobin and distorting the shape of your red blood cells. These deformed cells could look like this, sickled cell shaped. Okay, they're very fragile, very easily breaking. Okay, they reduce the number of red blood cells in your circulation, and it will lead to a deficiency we call the anemia, insufficient red blood cells, and they block or obstruct the blood flow in the capillaries and the very small blood vessels, causing severe pain and damage to your heart, brain, muscles, and kidneys. So now we are now moving to the new era of DNA cloning using a development of your recombinant DNA technology. We have what we call as your restriction enzymes, and it began in 1970s when the researchers are discovering this what we call as restriction enzyme. This mechanism was used by the bacteria to cut and inactivate the DNA of your invading viruses. Okay, they could be used to cut any organism's DNA at a specific nucleotide sequences, producing a reproducible set of fragments. We will be looking more into the details of these restriction enzymes in the future presentations. This one is your HindD3. Okay, restriction process results in the formation of your overhanging palindromic sticky ends. Okay, your HindD3 is a site-specific deoxyribonuclease restriction enzymes. Okay, they are isolated from your Haemophilus influenza, and it cleaves the DNA. This one is the DNA into this AGCTT in the presence of the cofactor magnesium via hydrolysis. So it breaks upon, okay, it cuts the DNA strand. We have also what we call as the vectors. Okay, when researchers discovered the ways to insert the DNA fragments which are produced by the action of restriction enzymes into the DNA molecules, we call them vectors, okay, that are forming recombinant DNA molecules. This strand here is your PBR322 plasmid, is one of the first plasmids widely used as your cloning vector. This one is your TCR, okay, this is your tetracycline resistance, AMPR, this one, your ampicillin resistance, your ORI, or what we call as your origin of replication. All of these will be discussed in the future presentation. Clones, okay, when they're transferred into bacterial cells, thousands of copies or clones of the combined vector and DNA fragments are produced during bacterial reproduction. And a big breakthrough in the genetics field is when this sheep here named Dolly, okay, on this picture on your right, born on July 1996 and died on 2003, was the first female domesticated sheep. Okay, also the first mammal cloned from an adult somatic cell using the process of what we call as your nuclear transfer. Amazing. Your genome, okay, your genome, often hear the Human Genome Project, okay, this collection of clones that represent the organism's genome, which is defined as a specific complete haploid DNA content of a certain organism. Okay? And they're also called genomic libraries. And genomic libraries are now available for hundreds of species. You can just check out, okay, your NHS, National Genetics and Genomic Education Center, and also your NCBI. You can just check the entire genome using the GenBank. Okay, this pie graph here containing the introns, your miscellaneous unique sequences, your miscellaneous heterochromatin segmental duplications, simple sequence repeats, DNA transposons, LTR retrotransposons, signs, and the lines, and protein coding genes. That's the genome. Recombinant DNA technology. It has not always and only accelerated pace of research, but they also gave rise to the biotechnology industry, and which has grown to become a major contributor in your US economy. So this is your PCR method. Your double-stranded DNA is heated to denature, okay, into 95 degrees Celsius to break the two chains apart. Then it is cooled to a temperature where the short pieces of DNA, we call them primers, can bind and anneal. Okay, these primers will be complementary to the sequences in your DNA template. Okay, and the DNA is then heated to a temperature, or let's say intermediate temperature, to allow the polymerase to build the DNA chain from the primer. So that's what we call as your extension. And these three processes are then repeated to produce large amounts of new double-stranded DNA. And they will repeat and repeat. So we call them recombinant DNA technology. So the development of recombinant DNA technology has emerged, and it has produced the development of biotechnology. Okay? In the US, biotechnology has quietly revolutionized, and it has many aspects of the everyday life. The products made by the biotechnology are very important and very famous in supermarket, in your healthcare, in agriculture, or even in your court system. So this example is brewing. It is an early application of biotechnology using the malt, okay, and fermentation process. Plants, animals, and food supplies has always using biotechnology. Okay, using the recombinant DNA technology, we could now modify crop plants to revolutionize agriculture. Genes for traits which are resistant to herbicides, insects, and genes for nutritional enhancements have already been produced into your crop plants, and the transfer of heritable traits across a species using recombinant DNA technology is what we call as your transgenic organisms, or your GMO. In medicine and genetics, biotechnology is also used because more than 10 million children in the or adults in the US suffer for some form of genetic disorders, and every childbearing couple faces an approximately 3% risk of having a child with a genetic anomaly. And the molecular basis for these hundreds of genetic disorders is now known in your OMIM, Online Mendelian Inheritance in Man. You can just check it out online. And these genes have been mapped, isolated, and even cloned. So this example, this photo on your right, is the example of a DNA profiling in order to determine the father of the child's CH. Okay? So your child's DNA sample is shown here, which is a mixture of a different sizes DNA bands of both the parents. In this case, the more similar to the child's DNA profile is this father number 1, which is likely to be the father. Next would be a human chromosome set. Okay, this human chromosome set showing the location of some genes whose mutant forms will cause hereditary diseases, and conditions that can be diagnosed using genetic testing are indicated by the red dot. So you can just zoom it in. All of these, most of the diseases found in every human chromosome number has already DNA tests currently available, and also it is being used in the Philippines, and we diagnose newborn screening using the blood sample from the newly born infant, we could just screen them for diseases like phenylketonuria and many more. So in this example, I am showing here your spinocerebellar ataxia, okay, which is found on your chromosome number 6. It destroys the nerves in the brain and spinal cord, which was resulting in some of the muscle control or ataxia. It has many types, but this is a progressive, degenerative, and a genetic diseases affecting your cerebellum. There are some modern approaches on how we understand gene function, and historically an approach referred to as what we call as your classical or forward genetics was very important for studying and understanding the gene function. And this forward genetic approach is determining the genetic basis which is responsible for a phenotype. And these forward genetics methods will begin with identifying the phenotype and find or creates model organisms which displaying the characteristic that has been studied. Okay, this approach, geneticists relied on the use of naturally occurring mutations or intentionally induced mutations using chemicals, X-rays, UV lights as examples, and they caused altered phenotypes in model organisms, working through the lab-intensive and time-consuming process of identifying the genes that caused these new phenotypes. So this is what these are your mutations. It could be silent, nonsense, and missense, conservative or non-conservative. So we will be looking at the phenotypes, okay, in the protein levels. So sometimes it could be producing a different amino acid, arginine and threonine, and it could also cause a missense or stop codon. Genetic studies also rely on the use of your model organisms. The first generation of model organisms in genetic analysis included letter A, your Mus musculus, okay, the mouse, and letter B, the fruit fly. Others, microbes have already been used as model organisms. For the letter A is the baker's yeast, the Saccharomyces cerevisiae, and the bacterium Escherichia coli. There are some, this table will show us model organisms and how they are being used to study human diseases. This is Escherichia coli for colon cancer and other cancers, Saccharomyces cerevisiae for cancers and Werner syndrome, Drosophila melanogaster for disorders of your nervous system and cancer, Synabhorditis elegans for the diabetes, Danio for cardiovascular diseases, and Mus musculus for Lesch-Nyland disease, cystic fibrosis, Fragile X syndrome, and many other diseases. And lastly, we live in the age of genetics. And in the next slide, I will just be showing you the timeline showing the development of genetics from Gregor Mendel's work in 1860s, working on pea plants, to the current era of genomics started in 2000s, and its many applications in research, medicine, and society. And having a sense of the history of discovery of genetics should provide us a very useful coursework in this framework as we go on along with the study of genetics. I hope that you've learned something today, and thank you so much for listening. Don't forget to like and subscribe this YouTube channel. Thank you, and goodbye.